Okay, let's go ahead and get started. Hello and welcome to today's live presentation. My name is Erica Fernaro and I'll be your host for today. This is the second webinar in our ASCLD Rapid DNA series. Today's presentation is brought to you by ASCLD and the Forensic Technology Center of Excellence. The Forensic Technology Center of Excellence at RTI International provides technology transfer and integration through reports, on-site training, and web-based seminars and events. These activities are archived for continued application or review. The American Society of Crime Lab Directors is a nonprofit professional society of crime lab directors and forensic science managers dedicated to providing excellence in forensic science through leadership and innovation. I do want to draw everyone's attention to the chat pod on the left-hand side of your screen. I'll move that around for everyone. This is going to be the best way to communicate with us throughout today's presentations. If you have any questions pertaining to the material presented, please feel free to submit those there, and we'll be sure to answer them during our Q&A session at the end. Also, if you happen to experience any technical issues, please feel free to submit those there as well, and we'll be happy to assist you and get you hooked back up. Okay, I'm going to switch everyone's layout now. And while that's loading, I'm going to go ahead and turn things over to Brady Mills, ASCLD President, for a few remarks.
Thank you, Erica. First, I'd like to welcome everybody to the second webinar in the ASCLD Rapid DNA series. I'd like to thank you, Erica, for all your hard work. We wouldn't be able to do this series without yourself. I'd also like to thank the other members of RTI for all the work that's done behind the scenes that allows us to present these webinars. And again, I'd just like to thank RTI for their continued support of the American Society of Crime Laboratory Directors. Looking at the registration list yesterday only confirms to me that rapid DNA is a very large topic within the forensic community. We have over 180 registered for this event, and it includes members of the forensic community as well as our clients, the attorneys and investigators. So I think Vince's portion of the talk today will give us a new idea on how the two groups can partner together to use this technology. I'd also like to welcome and point out that we have an international component to our registration list for this series, and it really just shows the power of this learning platform. While I have everybody on the line, I'd like to just point out that the call for abstracts is currently open for the upcoming ASCLD 2015 Symposium. The theme of our meeting is excellence in forensic leadership and how this translates to policy and practice. We have a number of areas of focus for our symposium, which include, but are not really limited to, management, leadership of the crime laboratories in the 21st century, technology, and quality and accreditation. Our symposium is held for 2015 in Washington, D.C. at the Marriott Woodland Park, and it is in April 26th through 30th. So please, if you'd like to submit an abstract, please take a look at our website for additional information. I'd like to introduce our moderator for today, Dr. Cecilia Kraus. Dr. Kraus is the director of the Palm Beach County Sheriff's Office Crime Laboratory and has been with the Sheriff's Office for over 22 years. During that time, the biology unit of the Sheriff's Office has investigated rapid DNA technology for almost nearly a decade now, through collaborations with Dr. Mathis at Berkeley University and later with Inogenics. During this time with Cecilia Kraus, the laboratory has also extensively presented and published microcapillary validation studies. So with that, I'd like to turn it over to Cecilia for our presentation today.
Thank you, Brady. And I'd like to welcome everyone as well. We have a full agenda today with tomes of important information. The presentations by Vince Figarelli and Gray Amick specifically address the practicalities of using rapid DNA in a working forensic and law enforcement environment. We're going to begin the webinar with Mr. Vincent Figarelli. Vince is the superintendent of the Arizona Department of Public Safety Scientific Analysis Bureau. He began his career as a criminalist with the department in 1990. His current position, Mr. Figarelli manages the 4 regional crime laboratories of the Arizona State Crime Laboratory System and helps establish forensic policy for the department and the Arizona criminal justice community. Mr. Figarelli will present on the Arizona Rapid DNA program, which trains officers in the use of rapid DNA instruments and the use of a standalone database for the development of investigative leads. Vince will be followed by our second presenter, Mr. Gray Amick. Gray Amick is the technical leader of the Richland County Sheriff's Department's DNA laboratory. He began his career as a criminalist with the South Carolina Law Enforcement Division, or SLED, in 1993, and in 2004 he was hired by the Richland County Sheriff's Department to establish their DNA laboratory. In his current position, Dr. Amick supervises and oversees the technical operations of the DNA laboratory. Dr. Amick will present on the Richland County Sheriff's Department's rapid DNA validation and workflow, which was implemented in January of 2014. We'll begin with Mr. Vincent Figarelli.
Thank you, Cecilia. Wait for the slides to load here. Yeah, thank you. As Cecilia said, I'm the superintendent and the system lab director for the Arizona DPS crime laboratory system. And I'm going to be going over the program that we established to allow officers to analyze samples in the field and compare them to a database of Arizona offenders and arrestees in hopes of identifying suspects who have committed crimes. So I get a lot of questions. What the heck are you doing? Fortunately, people usually don't curse at me. They ask nicely. Rapid DNA is new technology, and there are a lot of questions about it. Approximately 2 years ago, we committed to buying a couple of instruments and setting up this program, and it's a statewide program. As you'll see when I go through my slides, we're setting this up so we have a cadre of officers around the state who can collect samples and run them on the instruments and then search the database. The natural follow-up is, the follow-up question is usually, does the FBI know about this? These are two totally separate programs. If you saw the first webinar, the FBI is focusing on putting rapid instruments into the booking stations, and this is completely different. This is focusing on the forensic side and forensic samples and hopefully developing investigative leads for officers and investigators and helping to identify suspects. So I kind of divide our rapid DNA program into two distinct sections. The external one is the one that I'm going to be talking about here today, where we're training the officers to operate the instruments and then doing the search. We are, however, also working on some internal uses. As Gray will talk about in his presentation, there are obviously some uses inside the laboratory that these instruments can be used for, such as if you're a statewide agency or have a database unit, you can do hit confirmations. You can use them for arrestee and convicted offender samples now that the GlobalFiler Express kit has been approved for use, and we are actually working on validating that for use on particularly arrestee samples in order to get them into CODIS quicker. And then obviously forensic casework unknowns are another type internally, another type of sample that you can use these instruments for. So what this talk is, it's an overview of the external program. The training and the network setup and the use of the instruments. We set up a whole officer training program to train the officers. I'll be going over part of that and how the database system is set up and how they do searches and all the things around the external program. And what is it? Cost-benefit analysis. I'm actually working on this at the next ASCLD meeting. As Brady said, they're looking for abstracts. Well, this is probably going to be one of mine, a cost-benefit analysis, which actually is going to go beyond just how much the instrument costs and how much the analysis costs compared to doing it in the laboratory. It's also going to cover saved costs to the investigator or the investigating agency. We've already had several cases where agencies have identified suspects in serious violent crimes, and they have to such a point where they felt the need to do surveillance on these individuals. Well, by collecting a standard from them and having us run it in the rapid instrument and then comparing it to the original forensic unknown, that's done within 2 or 3 hours, so they do not have to do surveillance for 1 or 2 or 3 days while they wait for the laboratory analysis. And also, I'm not going to be going over the validation. That's a little bit more technical than I'm used to anymore. I'm long gone from the bench, so validation is another person's topic. If you have questions about the validation, you can certainly contact me, and I can put you in touch with our technical leader who can give you information about the validation process. So I break down the external uses into 4 distinct categories. Upload of arrestee or convicted offender profiles at booking stations or at intake. We are not concerned with that. That is the FBI's project. That's the first webinar, putting rapid instruments into booking stations. We are focused on these other 3, providing investigative leads, identification of victims. We've already used it for that. We had a serious burn victim. Some standards were obtained from their home. They were otherwise unidentifiable. The samples were run on the rapid instrument, and the victim was identified as the person that they suspected that she was. And then, as I said, identification of apprehended individuals, i.e. suspects, for comparison to forensic unknowns in which we already have a profile. And that's the whole, we have the suspect, we have them under surveillance. As quickly as we can get a profile to compare to the forensic unknown profile, the better it is for the agency, so they don't have to continue putting manpower on surveillance of an individual. So one of the big questions, why did we do this? Well, quickly providing investigative leads to law enforcement. That's the big thing. Like a lot of laboratories, we have a large backlog. It's actually a little bit lower than 4,000 at this point. It's more like 2,000. This is actually a presentation, the original presentation I gave at ASCLD back in May. So we've actually cleared out some of the property crimes. But there are a number of types. There are a number of types of cases, such as property crimes. The one that I usually use is blood at a point of entry of a break and enter. There's more than sufficient blood there if the perpetrator has bled to run some of that blood on the rapid instrument and save some for laboratory analysis at a later date. The other reason is that if we didn't do it, someone else would, particularly law enforcement agencies. They see this technology, they're told, hey, you can get a DNA profile in 90 minutes or 2 hours, and they wonder why we can't, as a laboratory, leverage this technology to their benefit and to help them get the ILs, to help them run standards and things like that. The drawback is, if you're a law enforcement agency, you don't have anything to compare it to. You would have to build a large database of your own to compare to before it's really useful, unless you're doing a direct benchtop comparison between 2 samples. And really, the reason I decided to move forward with this was that as a statewide agency, we could leverage the database of samples of offenders and arrestees for comparison. So this is an overview of our program and our network. We use the Inogenix Rapid Hit 200s, and they are placed in, well, actually right now they're placed in 2 places, Phoenix and Tucson, and Flagstaff should be in by the end of this calendar year, hopefully by January. And it's accessible to officers 24 hours a day, 7 days a week, and it's connected to our Arizona database, which is within the secure closed network of the laboratory. And the small pond software, which I'll describe briefly in a future slide, is what actually houses the Arizona database. And that database is synchronized with Estes. It's synchronized with the state CODIS database. And here's a diagram showing how that system works. On the left-hand side, you see CODIS, which synchronizes to several folders, which then move the profiles into the 4 circles or ponds. And on the right-hand side of the center rectangle, you see what's called the LEA pond, and that's the law enforcement agency pond. And from the right are the instruments that feed that network folder from the right that feeds the law enforcement pond. And then all the loops around the circles are a diagrammic representation of the searches that are being done. So as I said, the Rapid Hit 200 profiles are placed into the law enforcement pond by the officers who do the search. Samples in all the pooled ponds are searched against each other as the profiles are added. And this is an important thing. Profiles do not get copied or moved between ponds. And ponds are, if you go back and look, ponds are the circles inside the rectangle, and pools are actually the loops, are actually the ovals looping around the circles. Not my terminology, that's small ponds terminology. I always get confused and have to remind myself what a pond is. Pond is small, pool is big. So we obviously have flexible search control. All the settings that are in that, all the pools and ponds that are in that diagram are controllable by us. Since we set up the network, we can add additional ponds if necessary or decide that the searching algorithm needs to be changed. But the other thing is that you can link to other external ponds. If another agency or another region wanted to link to our ponds, our small ponds, then they could do so. And if they wanted to do that, then we would have to execute a memorandum of understanding or an intergovernmental agreement, some kind of contract to outline the parameters for doing the searching between the ponds. So, the instrument that we use, like I said, the Enogenix Rapid Hit 200 and the small pond software, those two form the core of the network. And then we also have a custom search interface that will return the investigative lead information when the officer does the search. For those of you who haven't seen it, this is the Rapid Hit 200. Slightly bigger than a bread box. Actually, it's I guess semi-portable. It now has handles on the side, so you can move it around a little bit easier. But even at that, I think it weighs about 300 pounds. So it's not exactly portable, but it's really got a small footprint when you consider maybe 3 feet of linear bench space needed. It's really not too big. The cost, $270,000 for the Rapid Hit 200 itself, and $1,750 for each set of cartridges. There are 2 cartridges. We are still using the PowerPlex Hot Start cartridge sets, and those are $1,750, which works out to $350 per sample for 5 samples. There is a bulk discount if we ramp up the use of it. Enogenix will give a bulk discount and a break on the cost the more that you buy. Here's an up close of the cartridges. The control cartridge has a positive and negative control and a ladder sample, and then the sample cartridge has slots for 4 samples. So essentially you can run 5 samples every time you do a run, and that's how the 1750 divided by 5 equals 350. That's how that works out. This is a view of the backside. You can see where the sample swab, typically they're swabs, sample swabs go in, and the chemistry is kind of worked out in the middle, and the PCR reaction is done at the bottom. And a control cartridge, as I said, has room for 1 sample, the ladder, and the 2 controls. This is just a diagram of how the extraction works. The reservoirs contain the extraction solution, which is moved up into the chamber where the swabs are, and then the ELISA cells are pumped from the wells back down into the bead chamber, which is preloaded with the beads. The ELISA solution is mixed with the beads, and the beads are engaged by the magnets, and then the ELISA solution is transferred to the waste receptacle. And then the beads get 2 washes with ethanol, and the wash solution is dumped into waste. Then it goes on to PCR, and the PCR premix is added to the PCR reaction chamber along with the sample. And then the thermal cycler goes through 30 cycles, which takes approximately 15 minutes. And then the electrophoresis is conducted on the instrument itself. Standard capillary electrophoresis, 15-second electroinjection at 4 kilovolts. Then flushed with buffer and electrophoresis occurs at 9 kilovolts for approximately 20 minutes. So this is Small Pond. This is the front page of Small Pond. This is actually a mock. You can see the ponds that are listed are not the ones that you saw in the diagram, so this is just kind of a test database that we had set up. But you can visualize the ponds. You can see how many samples or profiles are in each pond, and then you can go into the cases that are in Small Pond and look at them, and the officers can review the cases as they enter them. You can drill down even further with additional case information. And ultimately you end up with a matched list, which tells you your search profile with the profile that was found to match it. And then if you drill down even further, you get to see the two profiles that matched, and you can look at the various loci and the allele calls at each locus, and confirm that it is in fact a match. Once they have that information, the specimen information from the Small Pond database, the officer then takes it to this custom interface that was developed internally by us, and they enter the specimen ID in the upper left, hit enter, and they get back the information of the match with first name, middle name, last name, gender, all the demographic information for that individual. So we set up officer training because we don't want to just turn officers loose and say, hey, here's an instrument, go forth and use it. They actually need to have some training, and the training program as it stands today, we've done 3 training sessions and have about 33 officers trained at this point, 32 or 33. And it's a 4-day, probably going to expand it to 5 days, but right now it's a 4-day training program that covers everything from basic biology to basic forensic DNA analysis in an effort to prepare them for doing the analysis and doing the search, and then if they have to go and testify in court, testifying to what they did and have a basic understanding of how everything works. So this was the first class. We had 10 officers with 3 Rapid Hit 200s, 4-day training class, as I said. And they are told and trained to only analyze single source samples. And I think this is the key to using the Rapid Hit technology in the field, is that you got to keep it, you have to keep it simple. Single source samples such as blood or saliva, when you get a single source sample, a high quant single source sample, it is very easy to interpret it. They are trained that if they have a mixture, or they think they have a mixture, they are not to do the analysis, they are to submit it to the laboratory. So this is kind of a side benefit. In training these officers, we are giving very intensive DNA training to officers in various agencies. This is not just DPS officers, it's officers from agencies around the state. And they are then able to go and tell their coworkers, traditionally who in the laboratory, when we've had an officer call and go, hey, I've got this sample, it's a swab from a counter, can you analyze it? And we've had to try to talk them out of it because we know it's going to be a mixture if it's a swab from a bank counter or whatever. Now they have their peers who've gone through this training, who've seen some of these types of samples analyzed, and their peers are telling them, you know what, this isn't going to work, you're going to get a mixture, you're going to get a low-level mixture with 3, 4, 5 people in it, and we need to just not submit this type of sample. So we're focusing on these single source samples, and we're telling them that there has to be enough for follow-up at the laboratory. So here's a brief diagram of how the officer does what they do. They collect the sample, they type in the information for their case in Small Pond, and then they add the specimen to the Rapid Hit, run the Rapid Hit, then the profile is transferred back into Small Pond, and then the Small Pond software does the matching process and returns the match information to the officer. So here's a brief overview of the training that the officers went through. So we started them off with an understanding of basic biology, and then followed it up with an understanding of basic DNA analysis. As you can imagine, officers were absolutely thrilled about this. The first ones were kind of voluntold that they were going to go. I had asked for the best and brightest from my own agency. The first class was DPS, and sure enough, I got some really fantastic officers who were very bright and really, by the end, they're talking about pull-up, they're talking about electrophoretic conditions, or things like that you would never think you would hear coming out of an officer's mouth. And then an understanding of identification of appropriate samples for analysis. and with the final outcome being that they will complete the course qualified to use the Rapid Hit 200 and the database as an investigative tool to develop investigative leads. So here's just here's some slides of kind of what they went over. You know, thrilling stuff like cell membrane, cytoplasm, difference between a nucleus, what a mitochondria is, diagram of a cell, not your usual officer training material. Description of CODIS. They need to understand what CODIS is and the difference between CODIS and this program and the fact that they're separate, with the different indices in CODIS and how they correlate to what's in the small pond database. And then we teach them, you know, how to look at an electropherogram. You know, they have to be able to recognize that it's a single source. So being able to go locus by locus and looking at whether there's, you know, one or two peaks, ensuring that there are no more than 2 peaks, and then moving forward with putting it in the small pond database. So this was our first case. It was a one-vehicle accident with 2 unrestrained passengers with blood on an airbag. One of the individuals, one of the officers who went through the course, was actually one of our officers who does accident reconstructions and is a member of the Vehicular Crimes Unit. And he knew that based upon the kinetics of the accident, whoever's blood was on the airbag would have had to have been the person who was driving the car. So he collected a known from the suspect. And as you can see, this is the actual electropherogram for that. And this is the electropherogram for the airbag. It was blood from an airbag. He just did a small cutting and ran it in the instrument. And as you can see, they're clearly the same profile. There's a little bit of a blow-up of it. There's a suspect known. And the airbag. So getting very good response. This is the blood sample. More than enough DNA there, and there was more than enough on the airbag to do follow-up analysis in the laboratory. But what it did give the officer was the ability to say very quickly that this is my suspect. There were a couple of people in the car, but this is my suspect, and this is the person who we're going to charge. I think it was a DUI situation. So, future plans, additional training classes. We have done now 3 classes. We did 1 in April and 2 more in September. And I anticipate that we're probably going to do 1 class. Logistically, we keep the class to 10 people because with the number of instruments that you need and the practical runs that they do during the class, 10 is probably the most you can have where it's still manageable to use the instruments and do the runs that they need to do to practice. The second thing on our horizon here is doing a test case. Really, this is the same analysis that's done in the laboratory with the Rapid Hit instrument, and we're actually using the Rapid in the laboratory, but you get a DNA profile. It's the same kit that's used throughout the country for doing amplification, and really, I think at some point, this should stand alone when it goes into court, and that's what we're going to do is identify a good case to run as a test case through the court system. And then familial searching. Small Pond does have the capability to do familial searching, so we're working on that as well. And as I mentioned towards the beginning of the presentation, we are also working on validating for arrestees and offenders with the objective of getting arrestees into CODIS quicker. So that's it. Here's a list of all the folks who have worked on the project. A lot of them are my lab staff members and my boss, who's been very supportive, as well as my department, who has been very supportive in this program. And with that, I will hand things off to Gray Amick.
Thanks, Vince. Good to hear you again. Saw you at ISHE. Vince and I gave a similar tag team presentation at ISHE, so it's good to hear Vince again. I'm going to be talking about some of our validation with the Rapid Hit, as well as how it kind of is working into our workflow at Richland County Sheriff's Department. So to give you some context of where I am coming from, I've been involved in many validations in the past, and most recently the validations with this instrument. PowerPlex 16 we validated after we got the instrument in December and put it online at the end of January of this year, and then they came out pretty much right after we finished validation with a new kit, GlobalFiler Express. So we went ahead and validated that, put it online in July. So validation, as everybody may know, kind of has a pretty known prescription right now, and that is, according to the FBI's QAS 8.3.1, you have to do these things for internal validation. So you got to look at known non-probative samples, reproducibility and precision, sensitivity and stochastic studies, mixture study, and contamination assessment. Usually when you think of validation, you think of tons and tons of data, a refrigerator full of samples, but I'm here to tell you, if you look at this instrument and the speed of this instrument, with these kits, it can in theory be done in a very short amount of time. So if I break this validation down into runs on the instrument, and now I'm talking about runs with GlobalFiler, just to be clear, there's two chemistries available on the Intogenics instrument. One's PowerPlex 16. You can run 5 unknowns, and each run has a ladder, a positive and a negative control. The GlobalFiler kit will run 7 unknowns with a space for a ladder. The lab determines when and how they're going to run their positive, negative controls. So this validation design here on this slide, I'm talking about GlobalFiler Express. So with 7 runs per run, or 7 spots or slots per run, I can do this validation in theory in about 3 days. So the things I listed that the FBI is requiring are listed here. And these runs, you probably tack on 1 more day to compile some stuff, that is the precision numbers, the contamination assessment if you design your experiments correctly, and some stochastic effect studies. That equals, in theory, the validation for GlobalFiler on this instrument to be about 3 days worth of runs, a 1 day of number crunching, about 7 samples per run, which is about 56 slots. And the GlobalFiler costs $250 a sample. It's $100 cheaper per sample compared to the PowerPlex chemistry on the Intogenics instrument. So if you add that all up, that's about $14,000 to do the validation at the standard the retail price of the kits. That was in theory. So in reality, when I went and did the GlobalFiler validation, this is what I scribbled out to do, and in actuality, it took me about 3 months, about 13 runs, around 91 samples, and that's the price that I got for those number of samples. And the reason it took longer was when they switched from PowerPlex to GlobalFiler, they had some software upgrades, so we worked through some of that, so it kind of extended my validation. So I started thinking, well, how does that compare to other validations I've done at Richland County? Well, I'm just going to click through these like this. So when we started the laboratory in 2004, I did a identifier validation. It took about 5 months. I put that asterisk there because we were also renovating the laboratory, so it kind of got extended for that reason. In 2007, we did Y-Filer, 2008 Mini-Filer, and 2010 we put PowerPlex 16 online. But all of those took 3 months. It was just kind of our standard time. And then last summer we put the kit that we're currently, I guess is our workhorse right now, is PowerPlex Fusion online. That was in 2013. It took about 2 months. And then at the end of 2013, in December, we got the rapid hit instrument from Intogenics. And we got that online about a month. And I was kind of surprised at that because I was thinking it would take us about the same amount of time as other validations. But because the instrument runs so quickly, it runs, within 2 hours it can run the kit, well, that makes your validation take a lot shorter too. So about 1 month, and then the rapid hit GlobalFiler Express followed that, and like I said, the theory is 3 days, but it took about 3 months. So post-validation is the stuff that's actually takes more time and it's more of a pain. That is, you got to write your protocol. Our agency or our department allows us to basically issue a memo to amend the protocol as a kind of a stopgap before we have to do the entire protocol revision. You got to train people, which doesn't take long at all. It's really not that difficult to run. You got to obviously give these people a competency test, which in our case is an old proficiency test. Then you got to issue the memos and certificates, and then you're ready to go. So when I first gave this talk, I gave it at a the Southern Association of Forensic Science meeting, a SAS meeting, a local forensic meeting, and they wanted to hear about workflow. So that's the genesis of these slides, and I thought it was kind of interesting. I thought I'd go back to when I started DNA, which was in probably the mid to late 90s, and we were doing RIFLIP. So I said, let me start there. So the workflow for RFLP used to be, for the people here who remember that, it took around 7 months to get it took 4 or 6 months to get around 7 loci. So we did extractions of phenylchloroform and did ethanol precipitation. We had to run yield gels. We had to do digest and a digest gel. We had to hybridize and do all the southern blotting. And to get our results, we had to do autoradiograms and then crunch it all on a PC, putting the alleles in the bins, and then write the report. And at that time, there was no CODIS or no RFLP data into CODIS. So that workflow, we thought was great. This technology is terrific. Move ahead to early PCR, which is probably 1999, 2000. At the state lab, we validated Profiler, Cofiler in 1999. So the early PCR workflow was about a 7-day workflow in theory, and that's about 13 loci with those 2 kits to cover the core 13 for the FBI. So a similar workflow, you do your extraction, which is still phenylchloroform early on. You do a slot blot. Quantitation, you have to normalize or dilute your samples, and then so you know what to put in your amplification. At that time, it was 2 amplifications on like a 480 thermocycler. And then you had 2 platforms you could kind of choose, a capillary or a gel system, so the 310s or the FM bios. And then you wrote your report, and CODIS was around at the time, and you had eligibility and upload to do. So today, the common PCR workflow in our lab is the PowerPlex Fusion kit. It's a 24 loci kit, and it can be done in about a day. That is, you do your pre-treatment, then you have a magnetic bead extraction. We use easy ones. You do a real-time quantifiler. You do your normalization. You set up a 9700 thermocycler. You separate, detect everything on a multi-capillary system like a 3130 XL. You write your report, review, and your eligibility upload. So I said, after getting to this point, I said, well, how does the rapid hit fit into this? Well, if you stick a rapid hit in here, you can omit the pre-extraction, you can omit the quantitation, you can omit the normalization, and rapid hit takes care of the extraction, the amp, and the separation. So what you end up is, instead of 8 steps, you got 3 steps, and that is extraction, amp, and separate and detect is done by the rapid hit in 2 hours. Your interpretation and your eligibility stuff is still the same. So I have this slide kind of evolve from old methods, so I added the rapid hit to this slide. So it's kind of a summary of what I just said. In 2001, it took about a month trying to quantitate how long each of these methods takes. It took about 33 days when I tried to add it up. In 2012, which is, I guess, considered our current PCR, and these numbers are kind of representative of batches and how samples really move through the lab. But we don't do one case at a time unless it's a rush. So we do these, we do samples in batch, we do cases in batches. So in 2012, it takes about a week to do a batch, and there's your numbers. And then 2014 with the rapid hit, we're measuring it in basically a day. You can do a batch from beginning to write up and review and everything. So let me show you a little of the validation data. This is kind of what Vince was showing. The electropherograms look like any other electropherograms that come off of any genetic analyzer. They're FSA files. They can be crunched with gene mapper ID or IDX. It also can work with gene marker. This electropherogram on the left is a known NIST traceable 2391C SRM component A electropherogram, and the one on the right is a proficiency mixture sample from a proficiency test that's a non-probative case, used as a non-probative case. This is the GlobalFiler Express electropherogram, so it has the purple dye at the bottom. It has 3 different sex markers. It has a Y indel. It has amylogenin. Those are both real small in the green dye. And at the large side of the green dye, it has the DYS391. This is the precision data for the instrument. Again, just compiling the ladders, all the alleles at every ladder, taking the standard deviation, and then here's the average standard deviation at each locus, and multiplied by 3 to show you can precisely measure what comes off the instrument to create your plus or minus half base pair or 1 base pair bin. And then this slide is a slide, again, that's been built over the years with the different kits I've been running, and it shows the comparison of those kits. Now, the asterisk is important, I think, in this slide, in that it represents that when I did this study with the rapid hit instrument, I'm using pre-extracted DNA. So I have to take that pre-extracted DNA and pipette it onto a swab and put that into the instrument, which is not really representative of how this instrument, is performing with evidence samples or buccal swabs, because the DNA is already extracted. It's what we call naked DNA. So it doesn't perform well with pre-extracted DNA, which is not the normal sample you'd use anyway. So those numbers are low for that reason, and I learned that from talking to Intergenics and their experience with it. So what we ended up doing was kind of a sensitivity experiment that's kind of a real-time sensitivity experiment. Kind of what I've said before is, okay, if I know the sensitivity of this instrument is, say, a half a nanogram or 1 nanogram, that's all well and good, but really what the investigator wants to know is, will it work with, say, a cigarette butt, or will it work with a steering wheel swab, or will it work with, insert whatever type of evidence you're talking about. It's not going to make any difference to the investigator, or they're not going to know the answer. Does your swab or does your steering wheel have 1 nanogram of DNA on it? So we kind of did a real time or a real world sensitivity experiment and did several kind of different samples just to be able to say to an investigator, if they come along and ask, well, will it work on this type of evidence, we kind of will know the answer, and that's what the slide shows. All right, let me take a breath here. All right, so what are the pros and cons of this system that we've kind of put in place? Well, the cons or limitation are sensitivity. It's not as sensitive as PowerPlex Fusion. It works with limited touch samples. So you can see from this slide, we did do a steering wheel swab and a desk phone swab and a couple other touch samples that aren't on this slide, and we got good results. So it will work with some touch samples, but our usual path for touch DNA, burglaries and stuff, is going to be with our Fusion system. The other limitation is you can't enter evidence samples into CODIS yet, according to the QAS. And the cost is $250 a sample. That might be a limitation or con for some people, but if you could accurately quantitate or calculate the manpower figures as well as the consumables, then you make it come to that number with Fusion. It's difficult to do, or I have a difficult time doing it. The pros are it works with what I call zero samples. So if it's blood, semen, saliva, it's going to work. It's going to give you a profile. It's ease of use. It takes 5 minutes to set up. Its speed, it runs 2 hours. Runtime, 24 loci for 7 samples. It has an onboard database that can be used for immediate investigative information or QC information. And you can enter reference standards into CODIS in a modified rapid DNA context. So if you saw the first webinar, you heard this word modified rapid DNA analysis. Well, what that is basically running these instruments with these caveats, or running this instrument with these caveats. And that is you have to have the developmental validation done for the kit. Well, that's been published, and I give the reference here for the GlobalFiler kit for Integenics' instrument. The lab has to do an internal validation. The kit has to be INDIS approved. So the GlobalFiler Express kit from AB is an INDIS approved kit. You have to do manual interpretation. So there's no expert system here. That is, it's treated just like your 3130. The data that comes off of this instrument, the qualified analyst has to look at it, interpret it, and then they put that into CODIS based on eligibility. And the lab has to be compliant with all the QAS guidelines. So all those things are a check in our lab. And what we've done is with that system, we've run modified rapid hit on suspects to put in our suspect database. So the Richland County has entered around 69 suspects into our LDIS after being run with GlobalFiler on the rapid hit by Integenics. So from that, the reason how we got that number is we did a project that we wanted to run 100 suspects and put them in. So we ran the first phase of that. We did 50, which was about 7 runs. GeneMarker was the software handling the FSA files. We had it create batch files for importation. Then after we reviewed the data technically, administratively, and for eligibility, and then we uploaded that into LDIS through a CODIS terminal the way we would our other system, our 3130 Fusion data, and the hits are being generated. So the phase 2 of that was 50 additional swabs to get 100. Well, I kind of changed the plan about this and decided, why am I going to wait to run 50 more? What we've done here is basically incorporated this instrument into our workflow, and we've done that by taking or segregating suspect standards as the evidence and cases are being processed. We're segregating those to be run on this instrument. So, and actually this morning our technician Ashley Dixon was doing a processing run. She was able to segregate a number of suspects, and then when she got to the number 7, she went over, she started the instrument, ran the 7 suspects, in 2 hours had their profiles generated, and then this afternoon she'll finish processing the rest of that run. So when that run goes to the analyst for completion, all the suspect standards will have been run, electropherograms will have been generated, and that'll be given to the analyst along with the evidence for them to run. So basically all that, the suspects are already done in that case, and that it has the advantage of separating the standards in time and space and technology or instrumentation. So at the end of say we get 100, then I'll compile those numbers. So the database that I mentioned that's on board is actually a neat feature that was implemented in the upgrade. And what it can do is an immediate search of completed profiles. It gives you ability to add a sample from the completed run directly into the database if you like. And it also has the ability to import other databases. Its kind of published use is for QC or positive controls or and for investigative lead. So what it looks like, my next couple of slides are screenshots of the instrument after the run's completed. So in column A, it shows you kind of a grade of the sample itself. So the green check means it got a profile based on the parameters you set. The red X means it didn't get a profile for whatever reason, not enough DNA or whatever. And then the column B represents a database search. So what you're looking at is, for example, the top item, 14078271116.2, got a full profile in column A, and then column B is an X, didn't match anything in the database is what that means. So if you go down to the next to the last item, you can see that item 13 got a profile, column A, green check, and then it matched a person in the database immediately after the run. It matches it. So I can take that and use it for investigative information right away, or I can go ahead and put that in my CODIS LDIS and wait for that hit to come and handle it however the agency wants to deal with it. And then this is an a different run, but a similar example of a screenshot, and that is a positive control. So our controls are buccal swabs from us, our analyst, our technician. And so in the A column again is the actual profile grade, I guess. And then in the right column is the database search results. So you can see the positive control of my buccal swab matched me in the database. So if you were to, and this is a touch screen on the instrument, if you were to touch a green check in the A column, you would end up with basically a PDF of the electropherogram. So you can immediately look at the data in this manner to see what's going on. All right, so that's basically the nuts and bolts of it, and I'll go over one of our cases, and that was an attempted murder. So I had an investigator come to me with the idea that, oh, I have some evidence that might be good for a rapid hit. And what he had was a shoe and a pair of pants from the suspect in this case. And the suspect had basically was a burglary gone bad and ended up shooting the victim. The victim didn't die. That's why it was an attempted murder. So the victim gets away. The suspect later goes and changes clothes at a girlfriend's house and dumps his clothes. And the Sheriff's Department acquires that evidence and the victim standard and runs it on the this instrument in 2 hours. I'll come back to that. And gets basically a match between the blood off of the suspect's clothing and the victim standard. So this slide here is our run sheet for this instrument, and it kind of mirrors, just to give you an idea of how many samples you can run, it kind of mirrors the slots in the cartridges. So there's 2 cartridges. One has 4 slots. Though they both have 4 slots. One of them is taken up by a ladder in the second cartridge, I guess. So 7 unknowns total. And I like to show this slide because if you look on the left, it shows what looks like a partial profile. There's some alleles there, like D3, VWA, D8, even amylogenin, that didn't get labeled. And then I got what I would say a full profile on the electropherogram on the right. So this is I like to show this because when you run evidence, you don't get everything labeled, or you don't get a full profile every time. Now there's a reason that those didn't get labeled, and it has to do with its instrument's dynamic threshold generation, and which is probably a topic for another talk. But those peaks look definitely intense enough, but there's a reason why they weren't labeled. So the case outcome was that the DNA profile from the shoe and pants matched the victim, which we gave that information to the investigator, and the next day he was able to arrest the suspect. So in conclusion, the overall stats that from our agency are kind of as follows, and that is when we had when we were running PowerPlex, we ran about 29 PowerPlex runs, about 135 samples. We've run about 38 to 40 GlobalFiler Express runs, and that's about 266 samples. We've aided about 6 investigations. We got 69 suspects profiles into our LDIS from this Intogenics rapid hit instrument. We got 4 benchwork matches and 3 CODIS hits from the suspects so far, and we're still getting them trickling in, I guess. And with that, I think I'd like to thank Roy Swigert and Intogenics. It's been very instrumental in helping us get this going, and our chief, who basically was able to see the advantage of having this and was able to write the check to buy the instrument and put it on our bench. So thank you.
Thank you very much, Greg. I really appreciate it. That was a lot of information and very, very useful. It's nice to see this technology being used in the field. We have several questions from our audience today. The first one is for Vince. What resistance have you had from politicians or the ACLU or from the public for local database?
I really haven't had any back. Can you hear me? I'm back on. Right? Okay. We haven't really had any backlash. We get a lot of questions about what it is, but I think it's important to remember that it is essentially the same setup as CODIS. It's just a separate database, but it's really functioning the same as CODIS. No additional information is being put into it that's not already incorporated into CODIS.
And in conjunction with that question, or similar anyway, has there been any resistance from law enforcement officers to give their standards
No.
in the staff DNA profile database?
Yeah, the operators of the instrument are absolutely required. They are doing a higher level of sample handling than officers at a scene or crime scene technicians. So that's a requirement of participation in the program, is that they give a DNA sample for inclusion in the staff database. Yeah, for what it's worth, I would encourage any officer or crime scene technician to provide a sample, because anybody who is upstream in the process can certainly, as most of us who've worked in the lab know, it's very easy to contaminate samples. So I envision at some point that a lot more folks will be included in staff-type databases.
Thank you. The next question is for both Gray and Vince. Do you have an idea as to how many failed runs there are when you run a cartridge?
Yeah, that's an interesting question.
Gray?
That's not a metric that I really keep up with. I mean, I know there's been talk of success rate and whatnot with this instrument or the instruments that are out there, but at this point, that's not a validation bullet point for the FBI requirement, and it's not something that I'm measuring. I didn't measure that when I validated, my 3130. There will be samples that fail, just like on your other instrumentation. There will be runs that fail, just like on your other instrumentation. So I would answer that by saying that kind of a measurement, or that kind of metric, I think is the company's job before they put that out for the consumer. I mean, once I pay $200,000 for an instrument and start validating it, I'm not going to design an experiment to see if I can successfully get a profile and measure that. So my answer to that would be, we don't count, or use a success rate as a metric. We just know that it produces profiles just like our other technology, and we have some that fail sometimes, and sometimes just like any other, I guess, technology.
Yeah, and I guess the question came from 2 points. One is, does the manufacturer keep track so that those are replaced because they are so expensive and they didn't work? And the second part to that is, let me see, the troubleshooting the cartridge. I mean, you can troubleshoot the conventional equipment, but if you have a failed run,
Yeah,
it's really difficult to
that's a good
troubleshoot.
that is a good
Do you have
question.
any comments about that?
The Intergenics is, from my experience, has been extremely helpful with that scenario. So if I have a run that, say, just crashes for whatever reason, say the power goes out or there's some technical issue, I just basically let them know and they replace the cartridge. If there's, if it looks to be sample related, then obviously that's my issue. But yeah, Intergenics has been very, they quickly will replace a cartridge if you have a failure that's cartridge related or instrument related.
Yeah, and I would agree with that. The Intergenics has been very supportive.
Okay, thank you.
If there is a something cartridge based that happens, they'll replace the cartridge. Anecdotally, I'm going to say, out of the and this is just guessing, out of the 20 times that a use has been attempted, there's been a failure of some sort, maybe 2 times, but it's been something that's been recoverable. Like Grace said, we're not tracking failures, but I feel it's more than reliable enough in these instances where we're using it to go ahead and commit a sample to it or part of a sample to it.
Hey, and Cecilia, let me add one thing to that.
Okay, excellent.
You can,
if
you have a sample that fails, you can remove it from the cartridge and rerun it on the rapid hit or with other technology. So there's still DNA left on the swab if you need to rerun it.
Right, and we have actually done that.
Okay, fantastic. This next question is for Gray. Will this technology work on fresh DNA, or will it work only?
We haven't done like a
old and
study
dried
of
materials as well.
I guess aging material. But I'll take that back. The proficiency samples we ran as non-probatives were 2 to 3 years old. So that's how I'd answer that question. It has run in my hands on samples that are a couple years old. I don't have anything beyond that.
And by nature, our samples are usually fresh, so
okay.
Yeah, we don't have any info on that as well. Sorry, go ahead.
Oh, okay. No, that's okay. I'm glad you're adding this. Vince, this question is for you. Did you ever evaluate the rapid
No, we really didn't.
database?
I think that database exists to house the, or really is best used to house the profiles that are run on the rapid hit. Kind of early on, we recognized that there were some features of small pond with regard to controlling the searching and controlling search parameters that we committed to moving all unknown profiles over to small pond and doing any searching or evaluating of comparisons and doing matching in small pond.
Okay. Gray, this question is for you. Which cases in particular
the way we have it
are
set up
considered
is
rush
basically
court
if an investigator wants or thinks they want to run something on rapid hit, they have to go through our chief, who basically, buys the cartridges for us. So they have to convince him it's worthy of a rush. So that's kind of how we have it set up. We haven't had any kind of court order to do any of this work per se, but it's basically case by case, and if we agree it could be run with this instrument, it would have benefit the investigator, then we kind of get chief's approval.
Okay. The next question is for Vince. When undergoing laboratory accreditation assessments,
no, the program is designed and set up to be external of the laboratory operations, so it's not part of the laboratory and thereby not under ASCLD Lab. We're ASCLD Lab accredited, ASCLD Lab International, so it's not a part of our accreditation scope.
Yeah, the question was derived from I think the comment that you made that if there's a mixture and they need help.
Yeah, there is some reach back capability for evaluation of the sample as to whether or not it's a mixture or if they should move ahead.
Okay. And this question is for both of you. How do you determine, or if you're going to consume a sample, how do you do?
Well, we have,
documentation
you probably didn't notice in that worksheet, but we have a remaining column in that sheet, so we know right away whether we're using the last swab or not. And if we use the last swab, it's documented on that particular form.
And we wouldn't run that sample on the rapid.
Okay.
Through the rapid officer
Vince?
program. Yeah. One of my early slides that said something about there needs to be blood or saliva with more than enough sample for analysis in the lab, and if that's not the case, then it can't go through that program.
Okay. Gray, this question is for you. When manually reviewing profiles generated by the rapid hit, do you use any analytical and stochastic thresholds generated by the software?
What we did was basically adopted Intogenics' algorithm for the dynamic thresholds that they determine. So for this kit, and the data that comes off this instrument, we use a dynamic threshold that's set run to run for this instrument. We don't have a separate set analytical or stochastic threshold.
Okay. This question is for both of you, actually. As a matter of fact, Gray, I think you showed a slide that showed that there could potentially be manual review or override.
Well, what we do is
how often
we
do you
just
have
take
to do
those
that?
FSA files and look at them with conventional software. So we take what just like when GMapper makes a call or looks at an off-ladder peak, we have the same ability to look at that and agree or disagree with what the software is calling in that respect. Again, we don't quantitate or keep up with how many times we have to do that. The profiles coming off this instrument are very clean. I would even say they're cleaner than our Fusion profiles. And I think it's probably because the Fusion's very sensitive, but the profiles we get off this instrument have very, very little, few artifacts. So they have not a lot of analysts having to interfere or to look at or interpret additional peaks.
Thank you. I know that, Gray, you also showed a worksheet in which the knowns and unknowns were on the same run, and the questions for both Vince and Gray. Is this part of the procedure
what we
that
try to do
okay
is
to run the knowns and the unknowns?
If we have
do you
that
have any
scenario,
concerns
and
about
that's if it's a case like the case that I mentioned where the point is to get the investigative answer quickly, then and within 2 hours, you will have to run the knowns and unknowns together. And what we try to do is separate them on those cartridges. So we try to run the evidence on one cartridge and the standards on the opposite cartridge.
Which is exactly what we do as well. Yeah, same thing. The officers are trained that if they have a standard, they put it in the control cartridge that it's got that one slot left over, the fourth slot, and then the unknowns. And they do the unknowns first, just like you would in the laboratory. Handle the unknowns separately, put them in their cartridge, and then if you have a known, it goes in the control cartridge, and they're both done at a different time and handled at a different time.
Okay, thank you. And this question is for both of you as well. On what occasions will you have follow-up analysis in the laboratory?
No, you go first.
Right. We've done that on one case where we reanalyzed the evidence because we wanted to put it into CODIS. So we don't routinely have to do that, but on cases where we feel like it's going to benefit us to go ahead and run that evidence the other way so we can put that in CODIS, we'll do that.
And yeah, we require the officers to submit those cases to the laboratory. We've run approximately 30 cases, or the officers have run approximately 30 cases through the rapid hit instruments, but honestly, I'm not sure exactly how many have been submitted to the lab and how many we've done as a follow-up. But the objective is to, at this time, to follow up each one of them with the laboratory analysis.
Oh, okay. This question is for Gray. It has to do with your slide where you said that one of the steps has to do with our samples.
I've run
in which you noted them as being
actually
zero,
had
and you
case,
said
one of our
have you
first
done those
cases
studies?
was, and the one I just spoke of where we reran the sample, was actually liquid semen from a case that we ran through this instrument and was able to generate a mixture of victim and suspect with that case. So we have run semen, yes.
And we have as well.
We've run.
I'm
sorry. We've run.
Go ahead,
validation studies.
We've run that as a sample type, yes.
I'm sorry.
Yeah, yes, we actually have had a case where it was neat semen,
Okay,
and it provided,
Vince,
more than enough, obviously more than enough DNA for a full profile.
Okay. There was a question about your familial searching. Is that routinely done whenever you do a
well, we haven't had that set up
just
yet.
when asked, and how is that documented?
Yeah.
I think it,
Yeah. Oh, yeah. That's the future, and it's not set up yet. So,
okay, so
I'd love
your
to
future, then.
provide information, but we're not there yet. Now, I will add. I saw this as one of the questions, the familial question. Inagenics actually does have a paternity portion of software that they are going to release with the next revision. And I saw some comments in the chat about identification of in mass disasters with parents and children, or Katrina, and that software could be very helpful, as the person commented, helpful in those identification type situations.
The next question is for both of you. With regards to the cost of maintenance, if you don't use the instrument in a couple of weeks, do you still have to
there's a storage
run the system
cartridge you can
using
run
cartridges to keep it maintained?
that you run for that scenario. So you just pop it in, run it. It takes a couple minutes, and you're basically in storage mode till you run it again. Yeah, well, it's the same cartridge. It can be run, I think, 10 times, and it has
storage cartridge?
A set of consumables that go with it, but it's actually the same physical cartridge. Oh, I'm not sure. Basically, Inagenics gave us a kit.
And what's the cost of that?
And so I haven't had to buy one yet.
Okay.
Well, pretty much the same.
How often
we have some sample kits, and like Gray, I do not. We've actually bought some, and I don't recall how much they are. But they're obviously significantly cheaper than the sample kits. Like any instrument, it's it's good to keep them running, and if they're going to be down for a while, you need to use that storage kit. As far as maintenance goes, Inagenics has a field service team that can come out if there's any problem. We're not to a point where we have set up a maintenance agreement yet, so really not sure. Again, not sure how much that is going to be.
Okay. This question is specifically for Gray. It was noticed that on your precision slide that the number of samples or runs was 24, and you ran 13 for your validation.
Was a compilation of all
so
the ladders.
Are these all ladders for your precision? And
Oh, I see what you're asking.
So if there were 13 runs, there were 24
yeah,
ladders?
there was 13 runs.
I guess,
well, the instrument itself has a built-in file of ladders that can be used. So those are included in the calculation, because if the ladder fails on the run, it can select a ladder from a built-in pool of ladders to type the samples. I see, Cecilia. I see one that needs to answer. It says something about can you dump your local CODIS database into RapidHit to compare to samples? Absolutely you can. When I say you can import other databases, and we've done this, you can basically export your LDIS onto a thumb drive, stick it in this USB port on the front of this, and about 4,000 samples were imported into this instrument's onboard database within like 10 seconds. So that'll, of course, that's a snapshot of your database at the time of export, and so it gives you immediate investigative information. And another thing that, as far as CODIS, I would like to clear up is that you can enter samples, standard reference samples run on this instrument with the GlobalFiler chemistry into local or SDIS or any reference standard database, whether it's a RESTES, a local database, or say offenders. If you meet those modified RapidHit caveats, you can enter samples with the GlobalFiler chemistry into CODIS with this instrument.
And there was another question for Vince. When a suspect or victim elimination known is collected and compared to the
those types of samples aren't moved to small pond. If it's a benchtop comparison, they don't get moved. The only thing that gets moved from the RapidHit 200 into small pond are the forensic unknown samples.
Oh, okay.
And Gray, like I said, we did sensitivity
question regarding, do you know the lower limits
I
of
would estimate it's around probably 5 to 10-fold less sensitive than your regular STR techniques. So, you know, if you require about a half a nanogram, then it's probably going to be, you know, like I said, a little less sensitive than that.
Okay.
Okay, great. Well,
Erica,
if you do have any further questions, please feel free. You can email us. We will be sure to get them to the presenters and get those answered for you. Our email address is in the contact information down there, so please feel free to email us and let us know if you have any additional questions for our presenters. We do want to encourage you to visit your links of interest on the left-hand side of your page. It does have our short participant satisfactory link in it. It's also listed in your closing notes there. It does just take a few minutes, and we do appreciate any feedback that you can give us. Today's webinar will be archived, as we mentioned before, for your reference, and you all will be notified as soon as that archival does become available. As I mentioned earlier, also this was our second webinar in our Rapid DNA series, and you can find the first archival on the FTCOE website. We can also post that in the chat there for everyone. The next webinar will be on December 9th, and each of you will also receive a notification, since you have registered for this one, when the December 9th registration opens up and becomes available. You will receive an email on that. So it does look like that's all of our questions for today. RTI would like to thank ASCLD and NIJ again for their support in producing these webinars. A very special appreciation is extended to our presenters and our moderator for your involvement in today's webinar. And mostly we do want to thank all of you for your participation in today's webinar. We do look forward to having you in upcoming courses, and I hope everyone has a wonderful day.
I'm still here. I know. What are we? We're done.
You guys there?
Yeah, I'm still.
I'm still here too.
Yeah, I was going to sign off.